Good morning to one and all who have joined. Karthik sir, good morning.
Yeah, good morning, good morning. Just a minute, sir. I'm live streaming it.
Yeah. I was just checking my audio. Is it is clear
Yeah,
or
it's
not?
fine. It's fine, absolutely fine.
Okay, thank you.
Dr. Karthik Panikar, sir.
Yeah.
Sir, can you hear me?
Yeah, very much, madam. Good morning. Very much.
Okay. Good morning, sir. Thank you, sir.
Janjay sir, is Janjay sir there?
यस सर।
हाँ, संजय सर, आपको मैंने को होस्ट बना दिया, आप ऐड करते जाइए लोगों को।
हाँ ठीक है सर।
Karthik sir,
Yes, sir.
sir, I talked to Dr. Prashant Thackeray. He will be joining within 15 minutes. He's saying that I will join in 15 minutes because he's somewhere in the meeting. So
Yeah.
maybe delay by 15 minutes.
Okay.
And Dr. Mahendra Raut, I talked to him. He's joining within 5 minutes.
Okay.
Okay.
So we will start once Dr. Mahendra Rao joins us, or we'll wait for Dr. Karthik to come?
No, I think as Dr. Mahendra Raut joined, we will begin with our program so that within time the Dr. Thackeray will join.
No problem.
Actually, Dr. Raut is facing some problem. He will join within 1 or 2 minutes. Yeah, good morning, Dr. Mahendra. Hello. Dr. Mahendra, you are muted. Please unmute yourself.
Yeah, now you can hear me?
Yeah,
Okay.
very much. Yeah.
Okay. I'm
You are
sorry.
very-
I said I'm sorry because it's
It's okay. It's
very
okay.
early in the morning here.
Yeah, it's okay. And thank you very much for being with us. And it's
Thank you
very,
very much, sir, for inviting me. Yeah, thank you very much.
Actually, we all feel very proud and happy to see you, though it's online. But still, it's a very inspiring and motivational things for our student, to you people or being with us. Dr. Veena Mahajan and Dr. Karthik, sir.
Yes, sir. Yes.
Can you proceed now because Dr. Prashant Thakre?
Yeah, I mean, it is left to you. I mean, no issue.
Yeah. Dr. Veena, we can start with the introductory part.
Okay, sir.
Okay. And Dr. One more request from my side to Dr. Mahendra.
Yeah.
Sir, actually, what happened? Our one guest, Dr. Prashant Thakre, is from Amravati University. He is busy with some urgent meeting with the collectorate. So he asked me to join within 10 or 15 minutes. By some chances, he may be late. So in that case, you can start with your lecture also.
Yeah. Okay, that's fine.
Is
sir.
that okay to you?
Yeah, I think so. I think that should be fine. Yeah.
Okay. Thank you very much. Dr.
Thank
Veena,
you.
it's
Thank you,
Dr.
sir.
Veena. Okay. We can start now.
Okay, sir. Very good morning to one and all. On behalf of organizing committee, I, Dr. Veena Mahajan, Head Department of Zoology, J.M. Patel Art, Commerce and Science College, Bhandara, welcome you all in one-day webinar on biotechnology for sustainable development, jointly organized by Science Forum and Parent Teacher Association of J.M. Patel Art, Commerce and Science College, Bhandara, under DBT Star College scheme on the occasion of DBT Foundation Day, that is of 26th of February. Today we have with us our distinguishing guest. So it's my proud privilege to welcome very respected principal of our college, Dr. Vikas Dhumne sir. Resource person for today's technical session, Dr. Prashant Thakre, Associate Professor, Department of Biotechnology, Sant Gadge Baba, Amravati University, Amravati. Resource person for technical session 2, Dr. Mahendra Rao, Senior Postdoctoral Research Associate, University of Sheffield, UK. Professor Suman Deshpande, Coordinator, Parent Teacher Association, J.M. Patel College, Bhandara. Dr. Karthik Panikar, IQAC Coordinator, J.M. Patel College, Bhandara. Dr. Sham Dafre, Coordinator, DBT Star College Scheme, J.M. Patel College, Bhandara. All the heads of the department, faculties, and my dear students, it gives me an immense pleasure to extend a warm welcome to all of you. Biotechnology has become a pivotal tool for sustainable development in such a diverse areas as agriculture, environment, industry, and human health. It is an application of an explosion of biological knowledge, gives humankind the ability to alter the structure of life itself. One of the important aspects of biotechnology is its role in the sustainable development of various sectors. Sustainability is fast becoming the cornerstone of economy of many countries, both developed and developing. Such a development, in turn, depends upon the use of technologies, innovation, entrepreneurship, and utilization of an exhaustible supply of renewable resources. Biotechnology is one of such a technology which has rapidly developed over the past few decades and has great potential for solving many problems pertaining to agriculture, industry, environment, and health, which have direct relevance to sustainable development. These features and potential of biotechnology have generated great interest among the developing countries, many of which have embarked on various programs in biotechnology at various levels. To organize this webinar is an opportunity to discuss promising biotechnology applications that can contribute to sustainable development, as well as the need to develop capacity in science, technology, and innovations so that these solutions can reach their full potential. As the principal of our college, Dr. Vikas Dhumne sir is unable to join today's function because of his health issue, but sir, best wishes are always with us. Now, I invite Dr. S.W. Dafre, Coordinator DBT Star College Scheme of Chaim Patel College, Bandara, to give opening remarks. Dr. Dafre, sir.
Okay, thank you, Dr. Veena. It's a very pleasant morning, and I welcome all of you for this webinar, which we have been arranged for on the occasion of DBT Foundation Day. The chairperson of today's webinar, a very respected principal of our college, Dr. Vikas Dhumne. As because of his health issue, he is not with us online, but his wishes are always, and he always encourages to all faculty for having such programs. Even though during the pandemic situation, he's always encouraged and inspire us to organize such online conferences, webinars, etc. And we have arranged a lot many activities for students and faculties also during the last 2.5 years during the pandemic, corona pandemic situation. Guest speakers for today's webinar, Dr. Prashant Thakre is from Department of Biotechnology, Amravati University. He's busy somewhere in meeting, and very soon he will be with us for this webinar. And it's a very proud feeling to say one of the guest speaker for today's webinar is our proud alumni, Dr. Mahendra Raut. He's a student.
Thank you,
Dr. Mahendra, welcome for this webinar.
Thank you, sir. Thank you.
He is an alumni of our college, and he's a pass out of 92 batch. Am I right, Dr. Mahendra?
It's a 1919. Sorry, it's a 2000 batch. Sir.
Yeah, it's a pass out of 2000 batch. And let me apprise all of you, my dear student. He has a great success story because he belongs to a very small village besides Korombi. If you all people know Korombi, just to proceed onward to Korombi, you will find one small village, and he belongs to that village. And he came out of so many hurdles because I'm the witness of all these things. Being student, myself was a teacher at that time, and I know personally from his student life. So thank you very much, Dr. Mahendra, for being with us.
Thank you, sir. Thank you very much for such a kind word.
Dr. Kartik Panikar, IQC coordinator, he is always with us for the motivation and for arrangement of such technical issues. He is always behind us. All heads of the department, the coordinator of Parent Teachers Association, Dr. Sumant Deshpande sir, all heads of the department and faculty members, and my dear students. Actually, the reason behind to organize of such webinar on, especially on 26 February, now you people all of have an idea, today is the DBT Foundation Day. So DBT Foundation Day, actually, it has been organized. Let me aware all my dear students that in 1982, the government of India has established the National Biotechnological Board because the government of India was sought to improve the human health, agriculture, sanitation, and science and technology with respect to the clean energy. A huge, very wide spectrum of the vision of the government of India was there, and to overcome all these things, the government of India has formed, first of all, in 1982, the National Biotechnology Board. Thereafter, to come to specify the vision and the mission of the government in the field of health and etc. In 1986, especially the Department of Biotechnology has been formed. A separate unit to achieve the objective and goals of the in the field of human health, agriculture, irrigation, science and technology, equipment, startup program, everything. Huge area is covering nowadays by Department of Biotechnology, which is run by the central government of India. Now, DBT is instrumental in generating products, processes, and technology. It is also promoting biotech industry. It is also working in the socio-economic development. DBT always supports the productivity and cost effectiveness of agriculture, nutrition, environment, scientific, and the technological empowerment, you can say, of human resources. Beside that, many of we people, we do not know what exactly the DBT seek from the faculties and the student. Actually, DBT is running several teaching programs, several teacher training program. DBT is sponsoring the scholarship for research on the national and international level also. It sponsor a wide range of fellowship. It sponsor the international fellowship. It also sponsored the national and international awards, the popularization and promotion of biotech. It also deal with the translation of industrial development program. Mission program is there, including you people might have heard the mission, biopharma mission and biotech Kisan program. It is a new project which was launched, I think, in the it is 2021 by the Department of Biotechnology. So more than, and again, it you will come to know more than 20 to 22 autonomous institutes are governed by the Department of Biotechnology. Our college is also been awarded with the status of Star College scheme. Let me apprise Dr. Mahendra. In the 2 years back, the Department of Biotechnology Government India has given the scheme, has allocated us the scheme with the Star College scheme, and we have received a fund to improve our scientific infrastructure in the Department of Chemistry, Botany and Zoology. In the last 2 years, even there was a pandemic situation, we have arranged many programs for students and faculties under the Star College scheme. So this is and this is also part of the, and from the last week, from the 21st of February to 28th February, as 28th February is the Science Day, we are continuing, our science faculty is organizing various student-centric events. From 21st to 28th, every day there are some activities conducted by Science Forum under DBT Star College scheme in collaboration with the Student Parent Teachers Association. I think Dr. Thakre has joined us with us. Sir, very good morning to you.
Yes, very good morning, Mr. Shah.
Okay, sir. Okay, sir. Good. Okay, sir. Good morning and welcome to with us for being. I think with these words, I hand over the program to Dr. Veena because Dr. Thakre sir is with us. Okay, thank you very much.
Thank you, sir. Thank you so much for briefing us about the details of DBT. Thank you so much for your wonderful opening remark. I welcome Dr. Prashant Thakre sir on this webinar. Now, it's my proud privilege to introduce today's resource person for the technical session 1, Dr. Prashant Thakre. He is the Associate Professor in the Department of Biotechnology, Sant Gadge Baba Amravati University, Amravati. He is an ex-Senate member and he is a member on various academic bodies, like member of 11th Plan UGC Draft Proposal Committee for University and the member of 12th Plan UGC Draft Proposal Committee for Universities and many more committees. He worked as a treasurer and resource person in many national and international conferences. He worked as a nodal officer, COVID-19 Molecular Diagnosis Laboratory, Amravati. His area of research would be on molecular biology and also working on DNA barcoding of medicinally important plant species. Sir has published and presented many research papers in various reputed national and international journals. He has organized many workshops and related to technologies in DNA analysis, on microbial technologies, medicinal plants, etc. He has worked on 5 to 6 major and minor research projects and also work on international projects as a mentor. There are 2 patents to his credits, one for a kit for a rapid detection of Staphylococcus aureus, another for a method of rapid detection of biofilm irradiating concentration antibiotic against bacterial pathogens. Now, with this determination, now I invite Dr. Prashant Thakre, sir.
So, thank you, Dr. Mahajan, and thank you, Dr. Sham, Honorable Principal for the college, and staff and dear students. So, I merely congratulate you first for the Biotechnology Day, and I also congratulate the college for getting this approval for DNS Star College and conducting these programs earlier also. Today, when we talk about biotechnology, so I would like to share my presentations, but we'll have some remarks earlier also. When we are talking about biotechnology, what people think about it? When we talk about biotechnology, the people think about very sophisticated laboratories, some sophisticated equipments, people wearing aprons, micropipettes, the DNA, agarose gel electrophoresis and sequencers and like that sort of things. No, it's not the thing. What biotechnology comes for? Biotechnology is not a new thing. Biotechnology is always happening at our home. So when we talk about our home, what is that? Yes, indeed, our home, when we talk about this, biotechnology is there when you are forming your curd. Biotechnology is there when you are finding your breads, when you are brewing with the breads or cakes and all those things, when you are using with yeast and many more things which have come together in those things. So biotechnology, what came out? Biotechnology started a long back ago with the brewing of wines also. But now the biotechnology has become very common and it has boosted the industries like anything. We have biotechnology, which was market size which was valued about $752.88 billion in 2020 and is expected to expand at a compound annual growth rate of about 15.83% from 2021 to 2028. And this is by the favorable government initiatives with the growth of biotechnology in the sector in developing countries in India and China and all those things. I would like to share my presentations in those here. Okay. I hope you are able to see with this sharing.
Yeah, sir.
Yes, sir.
So biotechnology for sustainable development. Now, what is mean by sustainable development? When we talk about a sustainable development, that means the development should be there, but without any harm to the nature or to the environment. Actually, when we talk about this, there are a number of things comes through our mind. Is it possible? Actually, it is not that much very far possible, because if we so for development, we'll take an example of a development of a construction of a road. If you want to construct a road, then you have to remove some trees. So then again, we are damaging the environment in those cases. But development is an integrated part of the system that has to be there, but you have to develop without any damage to the environment in those cases. But where we stand in this COVID-19 era, Biotechnology was a term which was coined in 1917 by an Hungarian scientist, Karl Erck. He described the process to a large-scale production of substances from raw materials with the aids of living organisms. Or we can also call it as biotechnology. Defining biotechnology is exploitation of biological sciences to the technology. That is an integrated use of biochemistry, microbiology, and engineering sciences in order to achieve technological applications of microbes and cultured cells in those cases. So we have ancient biotechnology, classical biotechnology, and then modern biotechnology. So what was it? Ancient biotechnology, the brewing of wines from the grape, the formation of a curd from the lactobacillus. This was with an ancient biotechnology which was already there. Classical biotechnology came out with the finding of many things, such as the wine brewings, the developments in the wine brewings, plus the analysis of the disease based upon some classes, such as analysis of disease such as sickle cell anemia, or what we call it as sickle cell disease, based upon some genetic analysis methods. That was with classical biotechnology came up. And now we are in the era of modern biotechnology, where we have the third generation vaccines. Mostly we know that the vaccines which are getting today, such as with Covishield, then Moderna, and all those things. So these are the third generation vaccines or nucleic acid vaccines. These are coming out in the era of biotechnology here. So biotechnology with agriculture's biotechnology. So there are a number of fields where students can have their opportunity in those fields. If we talk about them, every time we don't require that we should be a part of the sophisticated laboratories. In the field of agriculture biotechnology in developing countries, we have plant tissue cultures for generation of virus-free clones. Banana is one of the good example in Maharashtra for this cultivation, where the virus-free clones are distributed or sold, and sugarcane is also one of the things. So which has revolutionized the agriculture sectors in those cases. Then we have another things which came up as BT cottons that we know. Medical biotechnology and numerous things which have come up here, that what type of things which are present is the vaccines, the diagnosis methods, etc., etc. We'll come to that particular points later on. Then coming up to environmental biotechnology, such as pollution clearing with bioremediation. Then we have coming up to the bio cleanups and all those things which come up in this. And then coming up to the genetic engineering, which helps up in all those fields, such as agriculture biotechnology, medical biotechnology, environmental biotechnology, also in food biotechnology. So when we are talking about agriculture biotechnology, so there are a number of things which are coming up, such as with plant biotechnology, then transgenic crops. Transgenic crops, like we have referred with BT cottons and all those things, genome edits, functional in. So number of things are there, molecular forming, etc., etc. We are coming up. We'll move to the next slide. The plant tissue cultures. We know with the plant tissue culture, I have given you an example of banana, because there are a number of industries in Maharashtra which are working on the plant tissue cultures for banana, also with the tissue culture fruits of citrus, pineapples, mangoes, etc. These are giving us, such as avocado is also one of the things, because nowadays people have become more cautious about their health. So avocados are also being used for plant tissue cultures. What is the yield of these things? What is the necessity of these things? Mostly we get those clones in the plant tissue cultures. These are same type of things, because they have been developed from a same type of plants. And when an apical blood is taken up for development into a sterile medium, it is practically free of any viruses, fungi, or bacteria. So whatever clone we get, they are extremely good and they are disease free. Then the quality of the plant that is chosen, They give you the same type of plants in all the fields. Almost excellent quality of the fruitings is observed in those cases. So this type of things have been observed with plant tissue culture, and plant tissue culture has been a successful thing for the generations of this. Actually, I would like to add to the story to this. Every plant cannot be used for plant tissue culture. The era of plant tissue culture, biotechnology in India started in 1982, when one of the former prime minister had been on a vacation in Goa. So he found out that there are a number of coconut trees which are growing on the seashore. And he asked, can we multiply them by any type of techniques? And an experiment was started that yes, we can try for it. But unfortunately, coconut did not give any tissue cultured plants, and that was a failure. But every failure starts for a success, and that was the point where we had started with the Department of Biotechnology in Government of Maharashtra, a separate entity than the Department of Science and Technology. So here, genetic engineering, it is an artificial manipulation of or alteration of the genes, also called as recombinant DNA technology or genetic modifications. It involves the removal of a target genes from one organisms and inserting that into the DNA of another organisms in those cases. There are many differences are there with these cases. In the recombinant DNA technology, what is revolutionized? We know with the number of persons which are suffering from type 2 diabetes. In this case, insulin-dependent diabetes mellitus, the pancreatic beta cells are unable to synthesize the required concentration of insulin. And older days, it was very well diagnosed that if the person is suffering from a diabetes and the concentration of the insulin was very low, despite the sugars are given up. So what an alternative came up? The alternative came up to extract the insulin from the pancreas of the calf means bachada, or from a pig. And this purified insulin was injected to human. There were two different things which were there. First was, it was the insulin from another source. And that type of insulin was giving an immunological reaction, because that was not because that insulin from pigs or calf was considered as a foreign protein. Second, sacrificing a pig or sacrificing a calf gave very small concentration of the insulin. That was not possible to do this. The story of insulin is very interesting in this case. A company named L. Lilly came forward and they produced the transgenic insulin, which was called as Humulin. Actually, we know that the functional insulin is made up of two chains, A and B chain. There was this A and B chain are held together by disulfide bonds. It was very difficult that both the chains to get multiply in the E. coli, because E. coli did not permitted the formation of disulfide linkages, and it were not being properly purified or synthesized in those cases. So two different organisms were cloned, one with A chain and one with B chain. After the purification of insulin in vitro, that is in glass, the insulin disulfide linkages were formed and it was marketed. This revolutionized the medical industries, that we can go for insulin productions. And slowly the production of insulin was also evolved. In Indian system, we have Wukat. That is a company which is present in Aurangabad, working in Aurangabad. They came out a few years back with a new type of insulin, which was termed as Lobastat. And this Lobastat produces the insulin both A and G in a single organism. So they some made manipulations in those cases also. So after insulin, number of things came up, somatostatin, then interferons, then growth hormones. So number of things came out after this. And E. coli was one of the bacteria which was used for all those things. Then gene cloning, the production of exact copy of a particular gene sequence in order to develop the line of genetically identified organisms which contains identical copies of the same DNA. So it was copied down and processed in number of organisms in those cases. Then we have transgenic animals and transgenic plants. So when we talk about transgenic plants, it's okay. Transgenic plants, we have transgenic plants in circulations. One, that is the BT cotton, that very well we know, because it is active as a biopesticide against the lepidopteran insects and cleptorian insects. Then we have the transgenic animals also. Now, what are those transgenic animals? Such as silk producing goats, sheep, rosy cows. Actually, they have been named under molecular farming. We don't have any permission in Indian system to use those. Only BT cotton and herbicide resistant soybeans or some other crops are used, but others are not used in those cases. Then we have gene therapy. Gene therapy is insertion of a functional genes into a cell or tissue or organ to correct the genetic abnormality or correcting a particular genes. Now, what happens with such humans? We have two sets of chromosomes. If you take an example of sickle cell anemia, if the parents, both of the parents are suffering from sickle cell anemia, then the child is obvious to suffer with sickle cell anemia. If only one parent is suffering and other is not, then the chances are 50-50, but the child will be a carrier, because most of the genetically inherited diseases are recessive in trait. So one normal gene is there to compensate the defect of another defective genes. Here in this case, if both of them are defective, the gene therapy is performed, an additional gene is added up to the system so that it will produce the normal functioning or produce the normal RBCs in those cases. So many other types of things are available. However, they are most costly even when we talk about type 2 diabetes or insulin-dependent diabetes mellitus. The beta cells of Langerhans, which produces insulin, if they are badly damaged, there are things that there are gene therapy is present that will introduce the genes into the system so that it will start producing the insulin in the body in response to the food intake. And they have shown it that it is almost 95% successful rate in those cases. Transgenic plants, when we are talking about, currently 16 countries permit the cultivation of transgenic plants. But in total acreage in 6 countries, 99% is there, such as USA, China, Argentina, Canada, Brazil, and Australia. 5 crops are currently released: soybean, cotton, corn, canola. Canola is an oil-producing plant. And then squash, tomato, potato, and flax in the past. Since the widespread release in 1996, the land devoted to transgenic plant has increased by 10 to 15% yearly. 90% of the Canadian canola is genetically modified, or mostly transgenic. 20 plus countries suspect the growing of transgenic plant without official approval. There are many things about 2 to 3 things I would like to explain in those cases, such as the tomatoes. What are those tomatoes? The flowers are. Everyone, we have bring tomatoes from the market, and if we keep it on shelf, they will ripen in 3 to 4 days. For delaying the ripening, an antisense technology was used in those cases. so that the shelf life was increased to about 10 to 12 days in those cases. So this is with one type of tomatoes which were available. Initially, when they were introduced in Western countries, the flowers were not accepted. Many organizations came against that genetically modified food. But then the government decided that we will have those foods packed up in two different ways. One is genetically modified and the other is non-genetically modified. The customer will have the liberty to choose which one they want. Then come up with the golden rice, which goes for the synthesis of vitamin A, the BT cotton, which is very well known to us. And then I have explained it about this herbicide resistant things. So here we can see the wild type tomatoes in 2 weeks, they are very good. But after 4 weeks, you will find that these are ripened up. But the flower side tomatoes, after 2 weeks, it's almost there. And after 4 weeks, you will find that they have been after as such as they are about 2 weeks. But Indian system, we don't have this to be permitted. They have applied for its production in India, but they not have been given yet the permissions because we started with COVID and everything was set aside. The golden rice, vitamin A deficiency is a major health problem. It causes blindness, influences severity of diarrhea and measles. More than 100 million children suffer from these problems. For many countries, the infrastructure doesn't exist to deliver the vitamin pills. The improved vitamin A content in widely consumed crop and is an alternative, attractive alternative in those cases. So this is the pathway which was there in those cases. We can see with phytogen synthesis and the beta carotene is synthesized, which is the vitamin A precursor. So this was taken up from the daffodils and was used in the rice. So the now rice produces the beta carotenes so that to compensate the deficiency of vitamin A in the children in those cases. So single bacterial gene for performing both the functions, and this golden rice has also applied in Indian system for in 2019, but now still they are waiting for its approval because number of genetic tests has to be conducted before its introduction into these systems. Then we have the Bacillus thuringiensis, that is insect resistant clause. I think most of you know about insect resistant cotton because this was the first genetically engineered product which was used for mass cultivation in the field of agriculture. But now we have many number of companies which have came forward for insect resistant cottons. But there is a regulatory authority in Indian system up to the district level, right from the national level to the district level, which goes on governing with this type of things. It happened with one of the companies in Gujarat. They brought the seeds prior. The Myco Monsanto conducted the trials in Indian system, and they distributed the seeds, and it was started for cultivations. Once it cultivated was there, they found that the growth was comparatively very good. The requirement of insecticide, that is chemical insecticide, was low in those cases. And then someone complained to this. So there was no option laid to remove those plants and to burn them because there was no permission for growth. But now Indian system, the Myco Monsanto was first given the permission, and now they have introduced number of companies are there which are manufacturing. Herbicide resistant crops that we have mentioned it here. Virus resistant papaya. We know with papaya. In papaya, we find that there is a spot comes up. Later, a small fungal growth appears on the skin of these things. This is, we call it as a ring spot virus in papaya. Indian system, we don't have any permission for the use of the transgenic papaya in which the viral coat protein was introduced so that there will be an immunological reactions against the ring spot virus. But the surrounding countries around India, such as Philippines, then Myanmar, Bangladesh, they are all cultivating. If we compare those things, the border is so porous that a person can move from India to Bangladesh and all those things. So we are still doubting that whether such type of seeds have been percolated into India and under cultivations. That needs to be investigated in those cases. BT brinjal. This was one of the things. We call it as vange or also called as eggplants. A 120-day crop requires almost about 40 to 50 sprays, chemical insecticide sprays for those things. So we can know how much amount of insecticide will be present in those cases. If you reduce it, then it will be about 5 to 8 sprays that are required for this portions. The BT brinjal was first developed with mycos by Dr. Bharat Char. I know him very well because he was a good friend of mine. Now have retired from these things. He struggled very lot for its permission to be given in Indian system for cultivation, because it is called as a poor food of the poor. But it was not permitted under the pressures of the organizations, NGOs, and all those things. Eggplant or brinjal is very well cultivated in Pakistan, Sri Lanka, Myanmar, Bangladesh, Philippines, Indonesia, and they are very well cultivating the BT brinjal. In Indian systems, the larger cultivation group is in Maharashtra, Karnataka, Gujarat, Madhya Pradesh, Bangladesh, Madhya Pradesh, and West Bengal also they are cultivating. So mostly, if you see with these things, one needs to investigate whether they have percolated their seeds into the Indian systems. Recombinant DNA technology was the first things which came or revolutionized these things. So most of them know with how it was cultivated in those things. We'll come to the environmental biotechnology. The environmental biotechnology is applied and used to study natural environments. It is concerned with the application of biotechnology to solve the problems in ecosystem. It is considered as a driving force for integrated environmental protection leading to sustainable developments, such as with bioremediation, bioaccumulation, biodegradation, bioleaching, biomethanations, and all those things where it can be applied as such. So bioremediation is a branch of biotechnology that employs the use of living organisms like microbes and bacteria for the removal of contaminants, pollutant, and toxins from soil, water, and other environments. So we know about Anand Chakravarti, who had developed with the organic Pseudomonas species, which was able to remove the hydrocarbons from the things. Okay. Then bioaccumulation is a gradual accumulation of the substances such as pesticides and other chemicals in an organisms. So rather than degradations, if that is coming up, it will be better to accumulate it in the particular species and then remove the species from those things. Medical biotechnology is one of the things in the era of COVID. This has much more focused than any other things. This is a branch of where that uses living cells or cell material research to research and then produce pharmaceuticals and diagnostic purpose. Pharmacology, gene therapy, stem cells, and tissue engineering, and all those things. We'll come to the latest parts, what will be there. It is in the service of coronavirus, vaccine discovery and productions. Vaccines are biological preparations that are produced using the science and tools of medical biotechnology field. Both organisms, living and dead, parts of them are used to produce vaccines in medical biotechnology. The era of vaccines started with rabies, where the live attenuated vaccines was used by Louis Pasteur for the treatment of rabies. Then again, it came out with measles. And in Indian system, we have a very good success upon smallpox, almost eradicated with the use of the vaccines in those cases. So there are number of things. The polio vaccine has also been used. The polio vaccine, which was used either intramuscular for earlier days, but now it is given as an oral polio vaccine. So what we call it as OPV, and it has a grand success for eradications of the polio. However, we have some cases which surface regularly in some areas, but they are very negligible. So we have a success rate of 99.99% in polios also. And we had recently, we are going from a pandemic. Pandemic means a disease condition which prevails or which crosses the boundaries of the country. And we have the SARS-CoV or COVID-19 pandemic, which is still going on. And that was a highly contagious infectious disease with a broad spectrum of symptoms, which are caused by a novel virus, which was now named as coronavirus 2, SARS coronavirus 2. When earlier days it was called as NCOV 2019, that is novel coronavirus 2019. So this was with, we'll slightly go with, because that isn't part of interest since it is going on. So it is coronavirus. It is divided into 4 genera. This class, alpha and beta primarily infects mammals, while gamma and delta infects with aves in those cases. To be very precise, there are 2 pandemics earlier also. One was SARS-CoV-1 pandemic, which took place in China and other related provinces. And then we had MERS, Middle East Respiratory Syndrome, which was caused by the coronavirus families in those cases. Here in those particular cases, MERS, it jumps from bats to camels and camels to humans. Such type of infections which are prevalent in animals, but when they are transferred to the human system, we term them as zoonotic infections. And there were many things which were going on about SARS-CoV-2 also, that is novel coronavirus, that it was lab made, and then for that, when we compare that of SARS-CoV-1 also in those cases. The SARS pandemic, which was there in 2003, and MERS, which were there. Now we have this particular things. What we have learned from this? Nothing. Is it came? Did it came? No. It is not the first time it came. This pandemic came up earlier also. Spanish flu, H1N1, it came out in 1918 and it lasted up to 1920. And during that particular time period, one could find that the peoples were wearing the mask also. The quarantine centers were also there. The peoples were admitted to the quarantine centers, or what we today call as COVID centers. The deaths were also there in those particular cases and were removed. So those things are known was Spanish flu. See, the scientific evolutions has to calculate with this. During that Spanish flu, no one knew that it was a viral disease. Everyone believed that it is. To that is the Russian flu, H2N2, then Asian flu, then Hong Kong flu, then we have swine flu, which is still persistent in those places, but we don't know about its persistent, how and where it will come up and when it will come up. So this is the COVID-19 timeline which came up. This is the thing which have came out, that a number of things came out. We just have in a very declined phase of the third wave of COVID-19. Most of things we have found in the second wave that there were a number of deaths which were there. Being working in as a nodal officer in the COVID-19 laboratory, molecular diagnosis laboratory, I have found that the cases increased rapidly in the February 2020, and Amravati, the spur of the second wave started. We were put under very heavy pressures for these things, and I have observed it that most of the people who got things with a mortality which was there up to 3 to 4%. 3 to 4% mortality is also very high, which is a reported one. So such type of things we have seen. How the waves came up. The first wave, it came up only of about Wuhan and some of the basic mutations which were taking place. Second wave did came out with the Delta variant and the Delta plus variant, ended with the Delta plus variant, which proved to be very deadly and the high mortality in those cases. Where we lacked, and what is the role of the biotechnology in those areas which came? Most of us have known, or we know what is PCR, and we have learned it. Some of them have used it. What is the PCR in those cases? The real-time PCR was not known to many of them. In Maharashtra, when we are talking, when it started with March, there were only 3 laboratories: Kasturba, Mumbai, NIV, Pune, and Indira Gandhi Government Medical College, Nagpur. These were the only 3 viral diseases regional laboratories which were able to use or diagnose with the coronavirus. But the impact was so high that the government had to open at each district level at least 1 laboratory. So what I want to say, those people who did not knew about PCR was ready to handle, were trained to handle in a very short span to work out with a real-time PCR, a more sophisticated version of a thermal cycler or PCR technology, where the detection and the amplification and the detection goes on simultaneously. So this all things were started very rapidly, and under pressure we developed a network of RT-PCR lab throughout the country in those cases, and now it is being used. When we talk about RT-PCR, even small children do talk about it, that if I want to move around, if I am suffering from something such as something cold or sore throat, I will have to conduct a RT-PCR test. So RT-PCR percolated to the bottom level. Where we failed? We failed with the sequencing. With the initiation of the second wave, we thought, before the initiation of the second wave, we thought that the wave has over, the pandemic has came to an end, but it was not so. The virus was still persisting in some of the corners in the country, and it was changing its face by the mutations. We were unable to track. How we were unable to track? During the first wave, UK used to sequence out 97% of the positive samples. In India, our rate was 5% because we had lagged with the technology. We had to set up with the RT-PCR labs first. Saving the lives and detection was the first aim. Slowly we started with sequencing, and now we are sequencing about, Maharashtra sequenced about 10 to 12,000 samples per month here. And that was one of the easy thing that we could control with Omicrons. But Omicron also was a weaker things, but we should not treat that this is a weak or came out with a weaker things in those cases. How this was to be eliminated? If the COVID-19 came out, it was to be eliminated. Most of the people came out with the concept of herd immunity. Herd immunity means a majority of the population which is exposed to the virus or bacterial disease, they will develop the antibodies against them. Here you could not use these things for or expose the community to the viruses, because there will be a highest mortality rate, around 60% to 50 to 60% mortality would have been there. And if you had exposed that much number of populations, nearly 25% of the population would have succumbed. So the herd immunity had to be brought so that the antibodies should be developed in the particular persons by means of vaccines. Now, what are those vaccines? The vaccines are those things which induces immunity against the foreign particles, that is the bacteria or viruses in those cases. So there are a number of type of vaccines which were used. We talked about from the start with Louis Pasteur, who used the vaccines against rabies, then with measles in those cases. There are some vaccines such as the first generation vaccines, second generation vaccines, and the third generation vaccines. What are first generation vaccines? So these are the first generation vaccines. Inactivated or killed vaccines, that means heat-killed vaccines, that viruses are taken up, microorganisms are taken up, they are killed using formalin so that there is no risk of the disease. Attenuated vaccines means what? Though vaccines are, the viruses or the bacterial systems are there, but they are unable to produce a disease, although they may imitate as a disease-causing agent such as viruses or bacteria. So this type of vaccines also were used. Or a subunit vaccine, this is a second generation vaccine. Subunit generation vaccines like a protein, hepatitis B antigen, what we call it as an Australia antigen, which is also given to the kids. So that is one of the second generation vaccines which is being used up. And we have the third generation vaccines, that is nucleic acid vaccines or non-replicating or replicating RNA vaccines which are there. So what we are using here is this type of two, that is non-replicating and replicating type of viruses, such as we are using it in Covishield, Sputniks. DNA-based vaccines we don't have, but DNA-based vaccines was introduced for Ebola hemorrhagic viruses, which has eruptions in northern African countries. RNA-based vaccines, such as Moderna vaccines, these are the third generation vaccines which are used for the systems. So now the genetic vaccines which are used for COVID-19, such as genetic variants, SARS-CoV variant which is used. This has, it can be a DNA or RNA which is used up. Our cells make the genetic and trigger the immune response against it. This is a low cost and fast to develop, need to be stored at lower temperatures such as -20 degrees centigrades. It has been approved in UK and for COVID-19. These are CureVax, Innova Pharmaceuticals have developed these things. Then inactivated vaccines. It contains killed SARS-CoV virus, whole virus which was killed up using beta-propion lactones and then to be used, such as influenza virus. Then we have in Indian system the Covaxin, which is 2 to 3 companies only which are producing this first generation vaccines or inactivated heat killed or chemical killed viruses to be used as a vaccines in those cases. Then attenuated vaccines, things that we have explained up, that is can trigger the immune system, but they cannot cause the illness in those cases. So here oral polio vaccine is also there. The live attenuated vaccines in clinical trials is there, that is what Codagenics, but these are not yet to be marketed in those cases. The protein vaccines, that is spike protein. The spike proteins of the SARS-CoV-2 has been cloned and the purified protein is used as a vaccines. So clinical trials are almost complete for Novavax and serum. Sanofi is being using with GSK, and this type of vaccines may be available in very near future, could be able to be administered for children also for their protections in those cases. So this is again the second generation vaccines which is there for SARS-CoV-2. The vaccine effectiveness which we see here, Pfizer, BioNTech, Moderna, Sputnik, AstraZeneca, they have an efficacy of about 62 to 70%, then 79%. The highest efficacy was shown for 95%. Now every company will come out with their things that showing that we have a very high efficacy in those cases. It also depends upon a person's physiology, how it defends on those cases. So these are the things which came out: real-time PCR, automated nucleic acid extractor, which were not seen by the most of the MSc going students also. But now they have become very used to handle this type of things and got a trained human circles. The biosafety, the biosecurity, which was the protocols which were developed by the Department of Biotechnology, were also being implemented at each district level for these things. So now the most of the workers or students know what is the PPE kit and what is the biosafety, what is biosecurity, how to dispose of the waste. Everything came level in this COVID-19. So this is what we come out. So we know that how the swabs are taken, the swabs are specimen is collected, the genomic material, that is RNA, is extracted, the reverse transcription PCR is done, RT-PCR is done, and then we get the results. If the amplification is there, then it is a positive. If no amplification, but to a particular site, what we term it disease that is collected in a database, there WHO has classified these things on 3 different types. One is variant of interest, one is variant of concerns, and another is variant of high consequences. So whenever we find a new mutation, it is submitted to them, and number of same type of mutations are there. They will consider it as it is a variant of interest because it is spreading into that particular area. But they do track the clinical manifestation of the persons who is suffering from them. It is, if it is getting cleared completely in a very soon, and it is responding to the vaccines. Of concerns is that it is respond to the treatment, but it escapes the mutations. It escapes the vaccine treatment, vaccinations also. Then they consider it as a variant of concerns. And variant of high consequence, it is not there yet been found in Indian system or global worldwide also. The variant of high consequences are those variants, they don't respond to the vaccines, they don't respond to the known level of treatment, and the mortality is very high. So such type of variant is not yet found in SARS-CoV-2 or in any other type of things in those cases. So thank you for your attention, and I will be highly obliged if you want, if you have any queries.
Sir, thank you very much. It's really very informative lecture. And I'm sure our student of microbiology and chemistry, botany, zoology, biology side, they are really get acquainted with the historical developments of biotechnology, which you have expressed in a very nice way. It's over to Dr. Veena.
Yes, sir. Thank you, sir. Thank you very much for sparing your valuable time from your busy schedule. Once again, thank you, sir, for your for briefing us about the various biotechnological areas such as agricultural biotechnology, medical biotechnology, environmental biotechnology, especially the plant tissue culture, and the most important sustainable biotechnology during the COVID-19 pandemic. Thank you once again, sir. Now, it is an extremely proud moment to introduce today's resource person for the technical session. Second, Dr. Mahendra Raut, sir. He's a proud alumni of our college during the year 1997 to 2002, having a microbiology group. The future belongs to those who believe in the beauty of their dreams. Sir's life has passed through very tough time, and sir, you have overcome all the hurdles in your life. So you are truly the inspirational source for all of us. Sir, he is a senior postdoctoral research associate, Department of Material Science and Engineering, the University of Sheffield, UK. He is the expert advisor in the University of Sheffield. He was employed as a project assistant on various projects funded by Government of India related to environmental issues, public health, waste to bioenergy generation. He has worked as a project assistant in Niri, Nagpur, also contractual research assistant at National Research Center for Citrus at Nagpur. Sir has worked as a senior quality controller in Haldiram food industry. He has received grants for his various projects from LBNET, BBSR network in NIBB and CYNET, BBSRC network in NIBB. He has done his PhD from University of Sheffield. He has published many research papers in reputed national and international journals. There is a patent to his credits. Now, with this determination, now I invite Dr. Mahendra Raut sir to deliver his talk on sustainable production of biofuels and biomaterials. Sir, please.
Thank you. Thank you very much, Mahajan Madam, for such a nice introduction. And and a special thanks to the professor for giving me this opportunity. I am really feeling honored to be a part of this webinar. I also want to thank Thakre sir. He took us through the history and the current to the current status of biotechnology. So starting with my presentation. So today's my presentation will be more about the technical part and about the research. So I normally say that my lecture is actually research lecture than the guest lecture. So yes, as you introduced me already, my name is Mahendra. I did my graduation from J.M. Patel College. So I have very special, emotions and attachment with the J.M. Patel College, especially because I feel that whatever I learn at that stage, I never learned it again back from anywhere actually. So that foundation was very, very important. That actually, developed the passion for the research in me. So thanks to all my sirs at J.M. Patel College actually. So and then, so I just want to go through my research experience. You can see my slides, yeah?
Yes, sir.
Everybody can see. Okay. So just want to go through my research experience as I started. I don't know whether it is because I can't see some of the slides, early slides, but hopefully you all can see that at the moment. So I started my journey from 2000 when I come out after my graduation. I've been to the Department of Microbiology at the University of Nagpur, and I did my master degree in microbiology. And then in 2002, I joined NIRI and worked as a project assistant. I worked on various projects during that time. Especially, I was very privileged to work on the project which was on cell purification property of river Bhagirathi in the Himalayan region, which was one of the best project and very interesting project for me, because I had a chance to go to Gomukh 3 times in my life, where actually the Ganga river originates. So my part was to analyze the water and sediment qualities, looking at the bacterial microbial communities. So in that project, we actually found that the bacteriophages are the viruses that kills the bacteria. They are abundantly present in the water of Ganga river. So then I join food industry, Haldiram food industry, where I worked as a quality controller. Then again, having interest in research, I again went back to NIRI in 2006 and again started the project. And at that time, I started working on various projects from waste to energy generation. Then to pursue my PhD, I've been to UK. I joined Chemical and Biological Engineering department, where I did my PhD on obligate anaerobes. So this is one of the thing I introduced in my career, which is very special because most of the people work on aerobic bacteria, but anaerobic bacteria, they are very hard to cultivate, and that's why most of the people don't try to use anaerobes in their study. But the anaerobes are very important because they are the best producer of biofuels and value-added products. So that's why I had an opportunity to set up the anaerobic microbiology lab in CBE at University of Sheffield. So my project was on exploiting a different obligate anaerobes at proteomic level for biofuel and value-added production. Then in 2015, I started working as a postdoctoral research associate. Again, I started working on various projects where I started exploiting syngas fermenting bacteria and also lignocellulose degrading and biofuel producing bacteria. Also, I started working on algal culturing. In 2020, I shift my research interest to host microbial interaction research area, which is more about the medical field, where I used to look at the different pathogens and host interactions and how we can and try to develop the interventions to prevent these infections in the human. And for that, I have to culture various tissues, human tissues, and looking at different, the interactions between bacteria and host cells. And then in 2021, I joined another group, which is very interesting group in medical science. We are so currently we are trying to develop the material that we can use for the biomedical applications. So this is my introduction. So I'm going to the next slide. As Thakre sir said, that we have the current need is to meet our needs without compromising the needs of the future. So in the simple word, I would like to say that, for example, the soil fertility, one of the issue at the moment in India. So, the soil fertility is going down because we are using chemical fertilizers. And so because of the population, we have to increase our, feed stocks. But we can't compromise with the nature because it affects it. It has a negative effects on nature. So like soil fertility, if we use chemical fertilizers, we will slowly, gradually decrease its natural fertility of the soil. And one time will come where we will have no option but to use only chemical fertilizers. And that is so actually we are compromising the nature for our current needs. That is one of the example that I would like to say here. So we have to preserve the nature. We have to preserve our sources for the future generation. Whatever we are breathing today, I think it's very visible that how pollution is affecting not only, and climate change affecting not only in India but all over the world. And so the goal, the ultimate aim of the sustainable goal is to how to handle these current challenges in environment, social aspect and economical aspects. So in 2015, Sustainable Development Goals has been established by the United Nations to make sure that everybody will be, live in a peace and prosperity by 2030. And that's why there is a worldwide, everywhere, we are trying to, a lot of research going on, how we can meet these requirements and how we can preserve our planet. So my research is about the sustainable production of biofuels and biomaterials. So this figure is itself illustrating my research background. So I'm exploiting different microbial systems at omics level, advanced omics level, to understand their biology, like syngas fermenting bacteria. This particular bacteria, they utilize gases like CO, CO2 from nature, and they convert it into the liquid biofuels. So we are trying to understand the biology of those microorganisms, so we can improve them with the help of system biology, synthetic biology approaches, and improve the biofuel production. Similarly, I'm also working on lignocellulose degradation, which is one of the most abundant biomass available in the nature, that we can convert it into the biofuels. And other things are algal biofuels. So I'm actually, being a scientist, I try to pitch every part of the waste to energy generation. So I'm working on most of the bacteria which are aerobic, anaerobic, algal, all kind of microbial system. I'm trying to exploit them and trying to understand their biology and how we can improve them for the biofuel generation. So that is my ultimate aim of my research. So let's start with sustainable biofuels. I'm going to talk about some general introduction about why we need biofuels, why we are talking about biofuels today, and how my research is correlating with this, how my research is correlating or pitching to the Sustainable Development Goals, that I'm going to discuss, and also I'm going to show some my results as well, research results. So why biofuels? The most important thing is why we are talking about biofuels, because the existing fossil fuels, they are exhausting very rapidly, and even some of the expertise, they already predicted that by 2050 we are going to run out of all the stocks we have. So we have to replace these petroleum based products, and we have to find out the alternative which is sustainable and renewable. So, and also we have to meet the energy demands, because energy demand is increasing every day, day by day, because of the population growth, and at the same time it is affecting our environment. You can see that now it is very visible, the pollution and greenhouse gas effects and climate change. You can see that. So what we need at the moment is, we need a low-cost technology that can give us a biofuel in a sustainable way, and also it should be a cost-effective. So my research is about to use the different microbial system and optimize them to produce the biofuels. So here we know about the different types of biofuels generation. So the biofuel means the fuel we can derive from the biological sources. So the first generation biofuel is actually the biofuel we can derive from the feed stocks. But the feed stocks is actually, we need it for the food. So we can't use it for the production of biofuels. If we use it for the biofuels production, then what we will eat? That is the problem. So it is not sustainable. So we come to the another second generation biofuel, where we can use the lignocellulosic biomass. So the lignocellulose is made up of the cellulose and other hemicellulose, this kind of polymers together. And these polymers are actually made up of glucose molecules or sugars, different sugars. That sugars we can utilize for the production of biofuels. Anyway, the waste, all the lignocellulosic material is actually it is a waste for us because we can't eat. So it's actually the waste, and it normally end up in the dumping sites and landfill sites. So we can use this material, the waste for the energy generation. The only problem with this material is that they are recalcitrant, that I'm going to talk on the next slide. So currently the biofuel market is growing bigger and better day by day, and it is projected that the global biofuel market will touch the figure of 200 billion USD by 2030, which is considerably very good growth. But at the same time, is it really enough? Because I think it's not enough, because the way we are running out of the fossil fuels, and also the population growth, the people's social economic status is also getting better day by day. So that ultimate demand of the energy is also increasing every day. So, and that's why we have to find out urgently alternative source, alternative to this to the energy, alternative energy source. So here you can see that I'm trying to show you the lignocellulosic biomass. They are made up of cellulose and hemicellulose, and they also made up of lignin. The lignin is also one of the part of lignocellulosic biomass, but we don't. I didn't show it here because lignin is more recalcitrant, and we can't use it as an energy source. Most of the bacteria, they don't use it. Some of the bacteria, they can use it as a source of energy, but most of them are don't use them as a source of energy. So we are mostly interested in cellulose and hemicellulose, which are actually made up of sugars. So these sugars we have to do. So the major problem with the use of lignocellulosic biomass in biofuel is that we can't use them directly for the biofuel production, because not every bacteria can degrade them, and not every bacteria can convert it into biofuels. So we have to treat it. So the treatment is we call it hydrolysis. In hydrolysis, we convert this lignocellulosic biomass into the simple sugars like glucose, arabinose, and xylose. These are the sugars. Actually, these are the monomers of these polymers. So we have to do the hydrolysis. Once it is converted into these sugars, then we can convert it into the the fermentation product. So the major challenges: recalcitrant nature of the polymer and the saccharification process. The saccharification means hydrolysis process, which is actually the bottleneck. If you don't solve the saccharification problem, then you can't convert it into the the products. So currently there are quite few techniques are in use to convert lignocellulosic biomass into sugar monomers. It includes physical and chemical techniques like steam explosion, acid and alkali treatment. But both of these techniques are actually energy intensive, and that's why they need a lot of input, and the output is very less. So it is not economically feasible. And that's why we have to look for the alternative which can give us a permanent solution and renew and continuous solution to this. And that's why we have to focus on biological system. So, but the biological system also has some advantages and disadvantages. So the delignifications and degree of polymerization of cellulose, they all actually so complex. These polymers are so complex that they are actually very very recalcitrant to these treatments. But the biological system can avoid this all chemicals and the use of chemicals for the hydrolysis process. So actually this process, the biological hydrolysis process, is actually mild treatment, we can say. But it has a disadvantages, like it is very slow at the moment. The treatment rate is also very low, not very effective for commercial application at the moment. And there is also the problem with the toxicity to the bacteria. So these are the advantages or disadvantages of the biological hydrolysis process. So the major problem why these disadvantages are there, because we don't know the systems, microbial system properly. There are millions and millions of microbial microbes in the nature, but we are actually playing with handful of the microbial systems. We don't use them. We didn't exploit them. We have the solution in nature, but we didn't found them yet. So we have to exploit them, and we have to make the microbial system better and efficient for the conversion process. So where here actually the synthetic biology can help to develop this biological systems. So synthetic biology is the tool that can fabricate or modify or manipulate the microbial systems and make them more productive and robust. So synthetic biology, in the current situation, synthetic biology is really doing well. It's providing a starting point to engineer the microbial systems, microbial strains for biofuel generation. It has some components like system biology components and metabolic engineering tools. The synthetic biology brings those tools together to make the system more efficient. So for example, we have waste gases and waste biomass that we can convert into biofuels, but we need the synthetic biology tools to make the system more robust and more efficient for biofuel production. So what is the but this is it easy? We can't do it. It's not like repairing the things, like putting nut bolt together and fixing the scooter or our machines. The biological system, in fact, are very complex, and they are very, very sophisticated, and that's why synthetic biology also has some limitations. So how we can how we can approach these limitations? And in that case, actually, the system biology approach like genomics, proteomics, metabolomics, these kind of approaches can help to understand the biology of the bacteria. So before going to the synthetic biology, we should know what this bacteria is, and then only we can modify the things. So like genomic approach will tell you about the entire genome of the system, and similarly proteomics will tell you about the entire proteomics of the system. And my research is always about the understanding the bacterial system at proteomics level, because proteomes are actually the key players and real players in the biological system. So say, for example, we say glycolysis process. All the steps are actually controlled by the enzymes. and these enzymes are the key players. So the understanding these enzymes is very important in order to develop the robust microbial system by genetic engineering. So here I'm trying to give you an example of the use of proteomics in my study. So I was working on one of the most efficient cellulose degrading bacteria. Cellulose degrading bacteria is Febbraury succinogen. This particular bacteria is found in the rumen of the ruminant animals, in the stomach of the ruminant animals. This bacteria is actually developed with the time. I think when the cow came into existence, I think the bacteria was already in her tummy, in its tummy. So you see the symbiotic association. So what this bacteria does actually, it degrades the cellulose into sugar molecules, and that sugar molecules utilized by the cow and other animals. So that in symbiotic association is very travel very long time, like I think it's a millions and millions years ago, that symbiotic relation is developed between the host animals and the microorganisms. So you can see here, this bacteria actually degrading cellulose. They are growing as a biofuels, you can see here. So the problem with this bacteria, this is very efficient bacteria, can degrade cellulose very efficiently in the natural system, but when it comes to the laboratory, it doesn't work in that way. So what is missing? So the problem is, we don't know this bacteria, how it degrades the cellulose, and that is where I thought I have to start working with to understand this bacteria. So I use the quantitative proteomics method to understand the biology of this bacteria and to find out what is the mechanism of cellulose degradation in this bacteria. And we found that it's a very novel cellulose degrading system this bacteria having. It doesn't utilize free enzyme system, doesn't utilize cellulosome based degradation system. It has a different novel enzyme system. So actually what this bacteria does, actually this bacteria grow as a biofilm, first of all, on the cellulose, and then they have different novel enzyme systems that and the adhesion proteins that adheres to the cellulose particles, and it is like a machinery, the complex cellulose degrading machinery which attach to the cellulose particles, and the enzymes, cellulose enzymes, they degrade the cellulose, and that is how the degraded products of this cellulose, they travel back into the microbial systems, and that is how this bacteria utilizes glucose and cellobiose from this derived from this cellulose. So this is another bacteria, Clostridium autoethnogenum, which is actually utilizing CO2 and CO, carbon monoxide, as a carbon source. So we don't need to feed the bacteria using glucose. We can feed it with the waste gases coming from from nature or from any like anaerobic digestion plant. We have methane, but we have CO2 gas as well. So that CO2 gas, we can convert it into a biofuel like ethanol, butanol, or 1,3-butanediol, like the advanced biofuels we can convert them into. So these are the pathways. When I use the proteomics approach, we actually come to know that which protein is regulating in what conditions. So if we have two conditions, like if we are growing our bacteria in glucose, and if we are growing our bacteria in CO2 waste gases, we have the different pathways to regulate, because they need different enzymes to utilize that substrate. And so the proteomics is actually tells you which pathways are regulated at that in the particular condition. You can see that I try to put the values here when the expression of the proteins. So based on the expression of proteins, you will know which protein are very important for particular synthetic biology approach. So from this slide, you can see that we found very novel enzyme system, which are enzymes, which are actually the key players in the cellulose degradation. So these key players, we can now give it to synthetic biology, and the synthetic biology can use that key enzymes for the constructing the novel pathway in a new bacteria. So that is how the different approaches, system biology approaches, helps synthetic biology to develop the robust microbial systems. So my research also on different, I use different bacteria, just like I said, I use Fibrobacter succinogens for cellulose degradation. Now I used Clostridium acetobutylicum for acetone, ethanol, and butanol fermentation. So this particular anaerobic bacteria utilizes different sugars and convert it into the ethanol, butanol, and acetone, different, the biofuels in a single fermenter. And so the production is actually very low if you use is at as it is. So we need to know why the production is so low. And that's why we use, again, I use the proteomics approach to understand its biology. And we found that due to the presence of lignin, when we use the lignocellulosic hydrolysate, due to the presence of lignin, it affects the proteomics, it affects the enzyme activity. Actually it reduces the enzyme activity. And that's why whenever we use the lignocellulosic hydrolysate for fermentation, the production of ethanol and butanol goes down. And this is because of the presence of lignin. And that's why it is very important to take out the lignin from the hydrolysate and then utilize as a for the fermentation. So that is another thing we found in our study. I also developed the co-culture fermentation process where I used two different bacteria. One is cellulose degrading and one is biofuel producing. Both the bacteria, I developed the media to grow them together to use the lignocellulosic hydrolysate and produce different biofuels. So with the developing this co-culture technique, we produce ethanol, butanol, and hydrogen in the same fermenter. My research is also about the anaerobic digestion, where I try to utilize the waste material, lignocellulosic waste material, to produce to improve the biogas production. Because traditionally we have biogas plants in India also, and we normally use the like we call it gobar gas plants. We normally use the cow dung and for this process. But nobody, I mean, there are so many scientists try to utilize lignocellulosic biomass for biogas production, but the production was always was very low, and it is very low because and it is because of the rate limiting step is the hydrolysis step. Because what usually people do that they use lignocellulosic biomass as such. But then I learned from the nature that in what happens in the cow's tummy, what actually the process, because when we use as such, if you put the lignocellulosic biomass directly into the plant, they will not, we can't convert it into the enough into the methane gas, because the hydrolysis is not done. So we need to optimize the hydrolysis process first. That is what I thought. And so in my study, what I did, that I tried to develop the consortia, like the consortia, microbial consortia, which is present in the human organisms, stomach of the human organisms. I tried to develop that consortia, artificial consortia, in the lab, and then utilize it for the hydrolysis process. And after hydrolysis, I use that hydrolyzed product for the anaerobic digestion. So you can see that the microbes are involved with time, and they have the ability to utilize different lignocellulosic materials. So they live in the symbiotic association. They help each other in this process. So as I said, in the stomach of the ruminant organism, animals, sorry, they have these microbial systems, and they are well adapted, and that's why they are very efficient to degrade the cellulose. And similarly, if you see the anaerobic digestion, when we use the anaerobic direct lignocellulosic waste in the anaerobic digestion, it doesn't convert into the efficiency is very low, and we don't get enough biogas. So as I said, we have the only problem is the lacking less efficient hydrolysis process and lack of adaptive microbial consortia. And that's why in my laboratory, I developed my artificial microbial consortia. I treat that lignocellulosic waste separately and then use that for the anaerobic digestion. And you can see the difference here. So when we use I use the barley straw biomass for this particular experiment. You can see that we produce a very less, the red, the orange color line showing you the biogas produced without microbial consortia and with microbial consortia. So the green line tells you how significantly we improve the anaerobic biogas production by this method. So coming to the next slide, which is now more about the biomedical sciences. So I have recently joined in 2021 a group where they try to utilize to produce different polymers, actually microbial polymers, the polymers that produced by the bacteria. We try to utilize those polymers in tissue culture engineering. Currently, we have different methods, to treat the infections and the diseases like heart disease and liver diseases. We have techniques. We have transplantation, grafting, all these methods, but all methods have some limitations. We can't, and they are very costly as well. So we have to find out a cheap alternative to these treatments, and that's why we try to develop the system where we can use the natural polymers to help treat the infected tissues in the human body. So as I said, I'm a fermentation person. So we actually produce a different kind of polymers in our laboratory. It includes polyhydroxyalkanoids. These are the polymers which have really nice properties that we can use in the human body. And we also produce bacterial cellulose. This is another thing, very interesting thing. The nature itself have ability to produce cellulose by microbes. We normally see that the cellulose is produced by the plants, but some of the bacteria are there that they can produce very nice pure form of cellulose. So we produce both of them in the laboratory. We harvest the bacterial biomass, and then we extract these polymers with the different steps. And once we extract these polymers, we purify them, and then we use them in a tissue culture engineering purpose. So here you can see that these are the polymers we normally produce in the laboratory. We combine these polymers with other materials like different nanoparticles or other biocompatible polymers to get the properties, because we have to match the properties of the property of the native tissues in the human body. And that property only we can obtain when we combine these polymers with other nanoparticles and all. So the to get the properties that matches to the tissue, human tissues. Then we construct the different scaffolds with the help of this help of these polymers, and you can see that these are the constructs we use. Now this construct then go to the we grow them. We try to understand how the human cells like cardiac cells, liver cells, how they helping, how they growing on this material. We test them in vitro, and then we once we found that they are compatible with the human system, then we use that polymers for in vivo in vivo studies, like in rats and things. And then once we approved once we got the positive result from there, then we go to the clinical trials and the further applications. So basically these polymers are actually work like patches. So suppose somebody has a hole in their heart, and you have to, because the hole is there because they don't, some of the cells are dead already in that particular part of heart. So we have to, how we can sieve it, or how we can patch it. So these materials actually work like patches. So we grow the cells on the top of this material, and then that material with the cells we can put it on the that affected area. And like I said, the cells, like if you have cardiac cells growing on this material, they will be stick to that particular part, and they will start growing there. And once they grow, that is how they will close that hole, or remove that hole. So this is the idea is to treat the various human diseases with this alternative technique, rather than using grafting and transplantation, because I think they have their own limitations and they are very costly processes. I also work on microbial biofilms and host tissue interactions, where I was looking at the pathogens, different pathogens. They infect human tissues, like the skin infection. You can see that most of the cases, Staphylococcus aureus, this kind of pathogens, they actually infecting human tissues. So my idea was to, because at the current in the current situation, the whole world is facing antimicrobial resistance, and that's why we have to develop the alternative interventions to control these infections in human body, human tissues. Because most of the time, whenever we because in UK itself, actually we stop using antibiotics. We only here the people they give antibiotics only when they needed, very in the worst time. Otherwise they actually stop using antibiotics here because of the antibiotic resistance developed by the pathogens. And so we need an alternative to the antibiotics, and that's why we tried in our laboratory to develop some of the interventions based on the peptide based which are actually we can produce in the lab. So these peptides actually interfere with the interaction between host cells and the bacterial cells, and that is how they stop the infections. So the idea is to develop the alternative to antibiotics. See, here you can see that there are some bacteria. You can see the bacterial cells in this figure. They are actually infecting the host cells. So these are the host cells. Host cells means I use the corneal cells, actually the eye cells, the corneal eye cells. So I use them as a host cells, and I I'm looking at how the bacteria infecting these cells. So they're actually infecting like a, growing like a biofilms. You can see here. So this slides, you can see the bacteria, the host cells are normal because they are not infected. But here you can see the cells are actually infected with the bacterial cells. So that is how bacteria eats, the human cells. So the idea is to develop the interventions, and this work is also under progress. So I would like to say thanks to Shyam sir. Really, I'm really feel honored to be a part of this webinar, because this is very close to my heart. My GM Patel College, really, and I'm really thankful to everyone. Actually, this is my first presentation I'm doing for India, and I would like to do this again and again.
Thank you very much, Mahendra,
Thank you very much, sir.
for being with us. It's really very proud moment that we are having two versatile personalities today with us. Dr. Prashant Thakre is a well-known figure in the field of science and technology and research, and he did a lot of very hectic work he did in the pandemic situations, first wave, second wave. It's really, we must salute his work, what he did in the pandemic study. He served for the society, and he really briefs the historical development of the biotechnology, what biotechnology really means. There is no need to go for taking courses and having a specific book of biotechnology. He really expressed a very fantastic way. The biotechnology begins with our kitchen and house. Sir, thank you very much. And Mahendra, yourself also, it's a very great work you are going to perform in the abroad. It's a
Thank you, sir.
Our students should must motivate and take inspiration from you, and be in touch with the institution and all our students and faculties also. So it's over to Veena to proceed further.
Yes. Thank you, sir. Thank you very much for sharing your valuable insights on biofuels, biomaterial, and its global importance. We rarely get this kind of opportunity to listen to the words of a great knowledgeable person like you, sir. Thank you. Thank you once again.
Thank you.
Now I invite Dr. Padmavati Rao, Head, Department of Botany, to propose vote of thanks. Ma'am, please unmute yourself. Rao, ma'am.
Yes, I already unmuted.
Dr.
Now
Mahendra,
you are audible.
Dr. Mahendra, you can take off your screen so that.
Oh, okay, okay, sorry.
Ma'am, can you hear me?
Yes, yes.
Sorry,
now you are
sorry,
audible, ma'am.
Okay.
Yeah, okay. I stopped staring. Sorry. It's because of me.
Thank you, Dr. Veena ma'am. Good afternoon to all. We had excellent webinar on the occasion of DBT Foundation Day on topic Biotechnology for Sustainable Development, for which our Honorable Principal, Dr. Vikash Domne sir, being Chief Organizer, today's resource persons, Dr. Prashant Thakre, Associate Professor, Department of Biotechnology, Sant Gadge Baba, Amravati University, and Dr. Mahendra Rao, Senior Postdoctoral Research Associate, University of Sheffield, UK, Dr. Shyam Dafre, DBT Coordinator, Dr. Kartik Panikar, IQAC Coordinator of our college, Professor Sumant Deshpande, Coordinator of Student Teacher Association, Dr. Ajay Gatole, Science Forum Coordinator, and teaching and non-teaching staff, and my dear students, welcome to all. It's my proud privilege to propose vote of thanks on this occasion. Words are not enough to express our sincere gratitude to today's guest speakers, Dr. Prashant Thakre and Dr. Mahendra Rao, who, despite their busy schedule, accepting our invitation to be the resource persons of this webinar, and guided us with their oration. Dr. Prashant Thakre has enlightened us with his knowledge and shared his research experience and views in the field of biotechnology. I must mention our deep sense of appreciation for his wonderful explanation about biotechnology for sustainable development in a lucid manner. Thank you very much, Dr. Thakre, sir. In the second technical session, Dr. Mahendra Raut has elaborated his research experience and also in detail about biofuels, biomaterials, which all of us could understand and might incorporate it in our teachings. Thank you very much, sir. I would like to thank our beloved principal, Dr. Vikas Domne, sir, for all words of wisdom and his ever significant continuous support and encouragement. I would like to thank Dr. Shyam Dafre, DBT Coordinator, Head Department of Chemistry, for organizing this webinar under Department of Biotechnology, Star College Scheme, Government of India. Thank you, sir. I am also thankful to Dr. Sumant Deshpande, Coordinator, Parents Students Association, Dr. Ajay Ghatwale, Coordinator, Science Forum, for their support, continuous support. And also thankful to Dr. Karthik Panikar, sir, IQAC Coordinator, for giving technical support for the success of this event. Last but not the least, I would like to thank Dr. Veena Mahajan for conducting the proceedings and also the organizing team of the webinar, teaching, non-teaching staff members, and the students of our college for making this event successful. Thank you very much. Once again, all the resource persons and the participants, thank you very much. And now I conclude this program. Over to Dr. Karthik Panikar, sir. Dr. Karthik Panikar.